Hello everyone, my name is Sergei Turanov and I am happy to greet you all, participants of the Urban Greenhouse Challenge Reforest. Today, I would like to tell you about the fundamental principles of interaction between light and biological system, for example, plant. This will help you to choose optimal light sources and develop light system for your future urban greenhouse. Let's get started. And the first question for today, what is biophotonics? Biophotonics is a discipline that studies the effects and interaction of photons in biological systems such as tissue and cells. In terms of plants, our main concern is radiation. Radiation is the emission or transmission of energy in the form of waves or particles through space or materials. But plants need special range of radiation for photosynthesis. Photosynthetic active radiation. It is the wavelength of visible light between 400 to 700 nanometers, in which organisms such as plants can transform light energy into chemical energy by photosynthesis. Do you want to know about the lumen output of a grow light? Don't even go there, because lumens are not important to plants. Lumens are for humans. The human eye is more sensitive to certain colors, and to account for this variation, scientists measure the light that humans see in lumens. Lumens take the amount of radiation and watts and adjust it to the human's eye sensitivity. Plants, however, use light from a different part of the spectrum, not necessarily the part that is measured in lumens. Let's take a look at the light spectrum with a curve showing what humans see. The human eye is more sensitive to green and yellow than to blue and red. 5 watts of green light has more lumens and appears brighter than 5 watts of red light. Let's look at what plants see. Plants clearly use light from different parts of the spectrum than what humans use. The human eye responds discount red and blue light, while the action spectrum shows that plants are receptive down to 400 nanometers and up to 700 nanometers. This is known as photosynthetically active radiation. The second statement is photosynthetic photon flux. Within the visible light, it tells us how many photons are emitted in terms of micromole per second. The third statement, photosynthetic photon flux density. Within the visible light, it tells us how many photons hit a surface in terms of micromole per square meters and second. And the last one, photoperiod. It is a 24-hour interval period during which a plant is exposed to light. What happens when plants interact with radiation? It's a very important question. If you understand one, you will choose the most effective light source for your urban greenhouse challenge. Let's have a closer look into a plant leaf. Let's see the efficiency of light interaction in a leaf. The incident light radiation is considered 100%. The majority of this energy, which is about 80%, is absorbed by the tissue. But only 2 to 5% will be used for photosynthesis. 35 to 40% will be transformed into heat, and one more 35 to 40% will be used for transpiration, the process of water movement through a plant. The energy loss will be as a consequence of reflectance and transmittance in and out of the leaf respectively. That is why the choice of light sources for plant is an extremely important task. Now I would like to describe you how plants can react for radiation. There are three main reactions, and the first of them is phototropism. In plants, it is a biological phenomenon which can be triggered by light by generating photoproducts. The plant receives the necessary stimuli about the surrounded environment. Have you ever noticed how plant turns to follow the light? How do flowers know to close at sunset and open at dawn? This is results of phototropism. The next reaction is biosynthesis. It's a complex chain of successive stages of the synthesis of organic molecules. A very good example of a chemical reaction that normally proceeds under the influence of light is the synthesis of chlorophyll. And the last of them is a lethal reaction. You can destroy plants if you choose incorrect light sources, for example, ultraviolet. It is a high energy radiation which damages plants. Well, we have studied quite a lot of useful information, and we can summarize the obtained data and to take a conclusion. There are three known roles of light in plants. Trophic role. This depends on photosynthetic photon flux, emission spectrum, and photoperiod. Regulatory role depends mainly on the spectral composition of the radiation. And biosynthetic role depends on the spectral composition of radiation and photosynthetic photon flux. Let's talk about different approaches on what light system to use. The first concept is based on the feasibility of using white light for growing plants. Why white light? It is simple. Plants evolved under solar radiation. Solar energy for us is mainly light and heat. The spectrum of solar radiation consists of 7% ultraviolet light, 32% visible light, and 51% infrared light. Most of the sun's surrealist energy occurs between 250 and 2500 nanometers. In this case, the power range in solar radiation contains the electromagnetic range from violet to red. Basically, white light imitates the spectrum of solar radiation in the power range. This concept has become very popular lately. The second one is focused on the amount of photons in green leaves. Green leaves consist of chlorophylls which absorb in blue and red regions of the spectrum. The rest of the radiation is reflected by the leaf, which is why it looks green. It is believed that illumination with only white light is useless, because this type of light has a lot of green and yellow wavelengths which will be simply reflected and therefore lost. In the last century, it was obtained a graph of the spectral sensitivity of a green leaf, and it is still used by many lamp manufacturers. And the third approach is using plants with known small range of wavelengths over time. This is the most popular concept among the scientific community. It has been proven that different plants contain different pigments, and therefore they might require different illumination. For example, in the morning some plants need more red light, when others seek for blue light. The ideal lighting device should control the radiation spectrum to provide enough light. This is one of the current challenges because researchers and engineers are looking for solutions to balance cost and performance. All three concepts have their own reasons, and at the moment there is not consensus within the scientific community. For example, TPU focus on plants that have a response to specific wavelengths. In each of your projects, choose the most effective and relevant light sources. And now, I would like to answer for one more important question. What photosensitive pigments are involved in the absorption of light by plants? Understanding this question will allow us to choose the right spectral composition of your lighting device. There are two main types of chlorophyll, chlorophyll A and chlorophyll B. Both absorption maxima are located in the blue and red areas. That is why magenta lamps are so popular today. But it should be understood that chlorophyll is surrounded by proteins. Proteins alter the absorption spectrum of chlorophyll. This is why magenta lamps are not as effective as we would like. Also, there are a large number of pigments that absorb in the green area and even ultraviolet radiation. For example, the phytoin pigment has an absorption maximum in the 270 nanometers region, and lycopene in the 520 nanometers region. So, that is more accurate to select the spectral characteristics. Also, there is a very important pigment phytochrome. It controls among other processes seed germination, flowering time and daily rhythm. Its absorption maximum is in the range of 660 to 720 nanometers. Thus, it can be seen that, depending on the combination of pigments in a plant, the spectral sensitivity of this plant can be very different. The first stage in the design of lighting devices for plants is the analysis of the irradiated plant. You need to know exactly what pigment the plant contains. It is advisable to study the condition under which plants grow in nature, and only after that proceed to design. How can this video be useful for your project? Let me explain this using one of the cases. Develop an automated lighting system. Light is an important component in growing any plants. As part of this task, it is needed to choose the most efficient location and the number of light devices, their shape and design, as well as offer light devices parameters. The first thing you do when you create a lighting system is the selection of radiation source. To complete this, analyze all the information this was given in this lecture and follow few steps. The first of them is select tree species which you want to grow in your urban greenhouse. Then, you need to search for information in literature and on the internet about leaves pigment content of the trees you selected. Well, you have studied all the information about trees. Sounds fantastic. So you can choose a light spectrum range of your future lighting device and decide the required photosynthetic photon flux density for your plants. And the last step, choose radiation sources which will provide the wanted light spectrum. I know, you have one important question: how to create your dream lighting device. Don't worry, we will explain you how to do that in the following video. Register on the website reforest.tpu.ru, create your team, participate and win. Together we will save the planet.